All right, ladies and gentlemen. In our last presentation, we were talking about homozygous, heterozygous, dominant traits, recessive traits. And now what we want to do is talk a little bit about how we interpret that information, what it means, and also a little bit about how we communicate about it and the tools we use to understand it. So what you see next to me is, what you see on the screen is a diagram. And what we have up here is we have two parents, okay? We have a pure green P parent and a pure yellow P parent. Now in this example, green is being represented by lowercase a and lowercase a, and yellow is being represented by capital A and capital A. Okay, so those are the alleles that we're using to represent those particular traits. Now, a pure green P, which you can see down here, when it creates gametes, it's going to cut its genes in half. It's going to make haploid cells. Those cells that were cut in half will only have one of the alleles. So you can see here, one of the alleles in this case is little a. The other allele in this case is little a. Now in this case, they're the same. They're not always the same, but in this case, they're the same. And the other parent, the yellow parent is going to make alleles that have one of its alleles as well. So big A, or in this case, big A. Now again, they are the same. They won't always be the same, but in this case, they will be. So what happens is when they make these gametes, we put the gametes on the side and the top of this box. And the gametes we put on the top of each column or next to each row. Because what we're doing is we're going to use this tool, which is known as a Punnett square, to determine the probable combinations that these two parents can make. So these two parents can only really make one combination. They can combine their capital A and their lowercase a, their capital A and their lowercase a, their capital A and their lowercase a, their capital A and their lowercase a. When you are writing the possible combinations, you always write the capital letter first. All right, that's just a convention. Now, you'll notice here that there's only one combination. So that means that 4 out of 4 are equal to big A, big A. Well, 4 out of 4 as a fraction also means 1. So there is a 100% chance that these two parents will have offspring that is capital A, lowercase a. That, again, is not always going to be the case. So let's actually take a look at an example that is not always the case. So let's say that we have that particular parent, big A, and they breed that parent with itself. So big A, crossed with big A, big A little a crossed with big A little a. So what's going to happen here is big A little a is going to give up either a big A or a little a. Big A little a is going to give up either a big A or a little a. So what we'll do is we'll draw ourselves a Punnett square. We will take these parents and put them on the top. So one possible gamete is big A, that goes over that column. One possible gamete is little a, that goes over that column. One possible gamete is big A, that goes next to that row. One possible gamete is little a, and that goes next to that row. Okay. Now what we use the Punnett square for is kind of like multiplication tables. We want to see how these things can combine. So here I take the A and I combine them right here. So I have capital A, capital A. So that's one possible way that the gametes from these parents could combine. Over here I'm going to take this capital A and this lowercase a, and that's another possible way that these gametes could combine. And then down here I'm going to do capital A and lowercase a, and that's another possible way that these gametes could combine. And then lowercase a and lowercase a. And that is a final possible way that these gametes could combine. Now, this is great. Now we have a tool that gives us all the possible combinations. But what we don't have yet is a way to interpret this. So what we're going to do most frequently is we're just going to look at out of 4, how many are each. So big A is 1 out of 4. Big A little a is 2 out of 4, which can be reduced to 1/2. Little a is 1 out of 4. Okay? So as we saw in our last presentation, these are all the genotypes. So 1 out of 4 or 25%. 2 out of 4, 1 out of 2 or 50%. 1 out of 4 or 25%. Those are the chances of having any of those allele combinations or any of those genotypes. Now, what's interesting here is the phenotypes, what the plant actually looks like, are fewer. So big A, and big A, little a both appear, in this case, yellow. And why do they appear yellow? They appear yellow because of dominance. The big A is able to hide the little a. All right? Now, also, you have little a, which appears green. So when you look at this chart, 3 out of the 4, or 75%, are yellow. 1 out of 4, or 25%, are green. Another way to look at this is 1, 2, 3. So if I was going to look at the green in relation to the yellow, for every 3 green plants, green seeded plants, I have 1 yellow seeded plant. If I was going to look at the genotypes, for every big A, I have so many big A, little a's, and so many little a's. So the way to look at this is, for every 1 big A, I have 2 little a, big A's, and 1 little a. And so that is how we represent the genotypes. And these two things are known as ratios. So what we have here are multiple ways to represent what we get in terms of the possible combinations, the probable outcomes. We could get a combination of genotype chances, which we represent as either fractions or percentages, a combination of phenotype chances, which we represent as either fractions or percentages, or we represent them as ratios. And the ratios for the genotypes and the phenotypes are almost always going to be different. All right, I think that's where we're going to stop for this presentation.